Hey everyone, this is Miss Moriarty, and I'm here to discuss with us the remaining parts of concept number 3. So, remember in class we started notes on 3 after everybody did their presentations today. Remember, the main focus of concept number 3 is looking at factors that influence the rate of photosynthesis. Um, Remember, in the first part of our notes, we talked about how the wavelength of light is going to impact the rate of photosynthesis, and how chlorophyll, which we know is the pigment found in chloroplasts, and other pigments, which can be found in a chromoplast, are going to absorb different wavelengths of light. And so, if we expose plants to those specific wavelengths, we may see a different rate of photosynthesis. Now, some other factors that influence the rate of photosynthesis was water. In our spinach leaf disc experiments, unfortunately, we couldn't necessarily test this factor, but we do know that water is going to impact the rate of photosynthesis because we know it's required in the light dependent reaction. We know water is obtained from the ground in the soil and carried up through the roots of our plant to then our chloroplast. If there is a shortage of water in the ground, we can see maybe photosynthesis rates slow down and maybe even stop. But don't forget, plants do have a cuticle, which we know has a waxy type feel or covering that tries to prevent that leaf from then losing any water. Another factor that's going to influence our photosynthesis rate is going to be temperature. Now, the reason is because photosynthesis is a chemical reaction, and we know enzymes are involved in chemical reactions. Enzymes are going to be influenced by temperature. So, enzymes tend to work best between 0 and 35 degrees Celsius. So, anything that falls above or below this range has the potential to then damage the enzyme. Right? Thinking back from the beginning of the year, we know enzymes have very specific shapes to fit with their substrates to then increase the rate of that chemical reaction. If temperatures go above the optimal temperature, that enzyme can end up denaturing or changing shape. When we see that happen at high temperatures, photosynthesis may stop entirely. At very low temperatures, we can see somewhat of the same result since we know at cold temperatures, enzymes don't work as fast. The last two factors that are going to influence photosynthesis rate will be then light intensity. So here on the graph, as you increase light intensity, it actually only increases to a certain point, and then photosynthesis actually then levels off. The same thing is seen when we expose plants to carbon dioxide concentrations. As we increase CO2 concentration, rate of photosynthesis will increase, but only to a certain point because they can only absorb so much carbon dioxide. And so when they hit that specific point, we see the rate of photosynthesis plateau or level off. All right, everybody, that is it for concept number 3. Please mark this video as done in classroom and work on your questions and summary for concept 3.